very nicely.
Okay, perfect. Thank you.
Anita, what is that, dear?
Hello. Hi, ma'am. How are you?
I'm
Nice
fine.
to
How
see
have you
you.
been?
Yes. Now,
Yeah,
Anita,
good, thank
I was
you.
wondering who is Anita. Now, looking at you, I can
You recognize
realize.
me?
Yes.
Right. I was yesterday a bit busy uploading Arogya, what do you call, Air Suvidha and whatnot, because I'm flying to India tomorrow. So, hello, Nakaseka ma'am. How are
Hello,
you?
Anita.
Nice to see you. So, I don't know, traveling is not easy anymore. It's also much paperwork. Yeah, it's the first time I'm going to carry a file to the airport.
And hopefully you can, through that file, get a smooth transfer. Otherwise, you are also going to be made to do some tests again coming back here.
Yeah, no, I mean, coming back to Germany is not a problem because I'm vaccinated. And so far, India is not in the high risk zone. I mean, Germany is. But going to India, then again, we have to get a PCR test done and wait for the results to come. The same old thing. But it's always good to be back to Amchi, Mumbai. You know, I'm just missing the vada paos and the street food. Although my bosses have told me don't eat any of that because don't fall sick. You need to come back. I said, no, that's my oxygen. I need it. I was missing it for the last 2 years.
Everybody is selfish. So he is selfish, you be selfish.
Exactly.
That is one big worry there, no? All the people.
Yeah. Actually, my boss is from Afghanistan. So it's not that he doesn't know Asia well, but he has lived in Germany for so long. But he was like, I don't think so. My body now adjusts to, the street food. And I'm like, no, that is, we are training our body every year. It's only because of COVID that we had to give a break. Actually, because of that, I even learned to make all these ragda patties at home. Because we were missing it. We are having withdrawal symptoms.
After all this thing, we finally about to start. Otherwise, there was some anxiety. Yeah. Also messaged me what happened and all that.
Yeah.
I just wanted you because that time difference, so I have to calculate exactly what time it is. So I didn't want to miss that. So that's why I thought I'll message you and say that I am online just to be safe otherwise.
Right. So thanks for joining in, and thanks Suresh also for agreeing to speak to our alumni.
Good afternoon, everyone.
Yeah, good afternoon. Thank you.
It's been a long time. I'm meeting Nagasankar madam.
Yeah, I'm also seeing you after a long time.
Very long.
I remember you in Toxicology lab.
Yeah, I'm going to discuss something about it.
Suresh, you were in Priya Gaonkar's batch, right?
Yeah, that's correct.
Yeah, because we have this SVBCP group. So yesterday after Mala ma'am said, so I put this, the flyer in that group and told distribute to your students. And Priya immediately wrote to me, oh, Suresh was in my batch. We studied together. And I said, oh, I didn't know that.
Right. So nice. It will be good catching up with everyone.
Yeah, absolutely. I mean, it's a bit nostalgic. Like even while making the slides, when I was downloading the BCP image, I was remembering everything, walking through the labs and all.
Right. Hope we can start soon. Yeah. Now we can start. I don't know how many people have joined or what is it, the other.
We will not come to know live stream mein kitne log hai. We will know here kitne log hai. Here there are 7 people only. 7 people only. Okay, only we are there. Only we are there.
So Suresh, where are you now?
Very much in Mulund, Mumbai.
Oh, you're in Mulund, okay.
He's with J&J, no?
J&J, okay.
He has that logo there, J&J ka.
Right.
So,
Yes, now I see. Yes.
You also know Suresh Surekha Prabhu. She was in QC and then she was in QC for quite a long time and then she left J&J.
Yeah.
She was, because we studied B.Pharm together in UD City.
Okay.
Live stream, what is the link? Then we can know from here, no?
I'm sharing the link. I'm sharing the link.
Acha, you're sharing it now. Anita, you're all set to come tomorrow, all other things.
Yes. Yesterday night, actually, I slept quite late. I packed my bags because we are planning to drop the bags today in the airport because the school vacation started from yesterday afternoon. So the airport will be full of people traveling for Christmas, and we saw that there are big, longer lines. So we are planning to put the bag now, check in and everything. So tomorrow we just walk through the security check. Okay. So that's the plan. I mean, yeah, I've never done so much planning to come to India.
So Anita, do you have to book for the test when you reach in India?
Yeah, I already did that. You have to do a prior test, and the rule is implemented from the 20th. And I'm landing on 20th morning, 1 o'clock. Yeah, so then we booked from here, but luckily you don't have to, you have two options. Either you pay before or you pay at the counter, but you have to take a token.
Okay, so you have to do booking.
Yeah, the booking has to be done. I think the reason is they want to see the influx of people and accordingly plan the number of test kits, what they have. Because probably it's happening that people come and then they don't have enough material to do the tests.
Yeah,
So just to get that idea. But it is also a bit challenging for people living abroad because they want an Indian telephone number and we don't have one. So they are like, if it's an Indian telephone number, they say it's an error. And if I use my father-in-law or mother-in-law's number, they send an OTP to them and they are sleeping in the night and we are like, oh God. So there was only one lab which was taking a German number. So we said, okay, let's book that.
Okay.
So it is, and
thereafter it is home quarantine, no?
Yes. 7 days home quarantine. And they said they'll call us 5 days, 5 times in a day. So I said, I will become a very good friend of that person if I talk to him 35 days in 7 days. And then on the 8th day, we have to take a test again to show that we are negative. Okay. And then we are allowed to go. But then I have another 7 more days and I have to fly back. My husband will be staying longer till the end of the month. But me and my son, we are flying back because he's having his 10th standard prelims. So he needs to be back here because his exams will start.
There it is 10th standard only, not 11?
Sorry?
The board is 10th standard also in Germany?
No, actually he is giving the Cambridge exam.
Okay.
So the international Cambridge exam, because in Germany, it is actually the 12th or the 13th. So it's like, they call it Abitur. So you either can do it, they call it G8 in 8 years or in 9 years. So the 8 years they had started 2 years back, and in Germany parents are like, don't
Only
stress.
India is not confusing, so all are confusing.
Don't stress the kids. They should enjoy studying. So now they've gone back to G9. So they give, it's like a 12th exam. So many Indian parents have a big issue when they come to Germany. They're like, they don't teach here. They are so like, they are not giving exams and all. Like, so they're not seeing that pressure in the kids. They all are like very cool.
That's nice, but
yeah, it's good. Like you see the children enjoy. Like I'm yet to see in Germany a child who is crying and going to school. Like they go so happily, like they're having nice times. So it's good to see that. But it's quite complicated, their education system, because they start splitting the kids at the 4th grade. So our 4th till 4th grade is basic education. And then based on how well the kid can perform, There are actually three tracks that they can go. One they call as gymnasium, which goes into university education. Then is something called Gesamtschule, which is a combination. So you can study some subjects in basic knowledge and some subject as higher, more advanced. And then there is something called Realschule, which is everything is standard education. So let's say you study about light, you will study only basic things about light. You won't study about particle theory or quantum theory or anything. So something like that. So I mean, ideally what they're showing is there is scope for every kind of child. But I feel splitting them at the 4th grade is too early because they're still learning, like how will they know what and how. But I was a late bloomer. I never liked to study when I was in school. I was only doing sports. So
with due permission, there are only 15 people now I see, and 9 of us are here. So I think we should start now because kuch gadbad hai to I'll follow up now with others. And let us, I am just master of ceremony. I'll be starting the meeting, and I'm happy that this Chai Pe Charcha is continuing every bi-monthly. We are doing it, and even the videos, whatever we are recording, we are keeping it in the Alumni Association. Yeah.
A quick request. When you send the flyer for Chai Pe Charcha, no? This time we got a little PDF document.
Yeah, that is the problem. I also just now noted.
You can't cut and paste the link. So actually, I was lucky that you sent the link 2 minutes ago, so I could join. Otherwise, it's kind of difficult if you're with a mobile to kind of note that and kind of join. So
and that too also, if you open it, the name and all this comes under the band, so it is very difficult to read the number and enter the number.
Yeah, so just maybe that's the reason why people are thinking that I have to note down all of these things on a piece of paper.
Yes.
And if they are standing somewhere, they may not be able to do.
Okay, so,
thank you.
I have put it now in alumni group also. I'll put it in the IPA group also.
Yeah.
And other groups also I put it. So thanks for joining. But student group में वो भेजा है कि नहीं मालूम नहीं है मेरे को. So that somebody will have to take care. Dr. Krishna will know whom to tell to circulate.
As this program is recorded, it will be circulated.
I will send you the link also separately, Dr. Krishna. Give me a minute only.
Can I take this opportunity to say a quick hi to both the speakers? Hi, Anita.
Nice to see
you. Hi, P. Suresh.
Krishna sir, how are you?
Yeah. I can't see you, Krishna.
You want to see me? Why, yaar?
Come on.
There you go.
There you are. Nice to see you.
All gone.
That's because you have so much inside.
Nothing is there.
It's always a pleasure to see you. How are you doing?
Very fine.
All well at home, family?
Yeah, all well at home. Admissions are going on, so we are in college.
Okay.
Late admissions this year because of COVID.
Okay.
What was supposed to occur in July, August
is happening in December. Okay. But then how is it? Then you'll have a complete year, or it will be a small consolidated year?
Because it's online and there are no practicals, you're going to kind of crunch everything quickly.
Okay.
We might have to forego hands-on practicals, give them theoretical.
Okay.
So it will be more lectures, and that helps in shortening it.
Okay.
But then, what is the 4-hour practical is done in about an hour because you give only the
theory part.
Yeah.
Okay. Yeah. And how is it over the years to your, I mean, of course, the final year, it will be a bit difficult, but let's say first year and all, then over the years, your plan to then expose them more to the practical part, because that is also important, right? Those skills.
Yeah, but what is happening now is since the last year and a half, 2 years, they're just not allowing us to get students. And it's a very catchy situation. They're telling, try and run it at 50%. But in the end, they have left one option open for the student to want to come or not. So, so I have a very odd situation. I have a class of 60, 40 might say I'll come, 20 say I won't.
20,
So
I
how will
won't.
I handle both of them?
Exactly.
Plus that 20 have to do the lab with a 6 feet distance.
Oh, okay.
Doctor,
Because
Dr. Krishnan, can we start, because we are already 25 minutes late, and I was also now circulating the things. So I can see some more people joining, and hopefully with the students also, they didn't have this, because I got a message from students also. Sir, the link, those students don't have the link. So I told Swati also to send a link to students. If you can send, you can also send. Good afternoon to all of you. And I welcome on behalf of BCP Alumni Association and our chairperson Parizad and our whole committee member for this Chai Pe Charcha. And I'm happy that you could take out time and you feel proud that yes, we should talk for and guide the students and alumni for BCP. So welcome Dr. Anita and Dr. Suresh. Welcome to you also. Before we start, we'll just pay the homage to our martyrs whom we lost, our CDC, Bipin Rawat ji, and the whole team members. So that is needed. That's what I felt. And we just remember them before we start the program. We can start the program, and the first speaker will be Dr. Anita. Next speaker will be Dr. Suresh. So first, before we start our program, Chai Pe Charcha, I will request our President to have 2 words before we go ahead with the program. Dr. Parizad, over to you.
Thank you, Ashish. A very good evening to everyone. Serving as president of our Bombay College of Pharmacy Alumni Association, it's an honor for me to welcome you all to this December session of Chai Pe Charcha. I appreciate this idea of this alumni meet, which creates a special bonding between the students who have passed out and established themselves in the outside world. This is, I believe, a perfect platform for you all to meet your peers and revive the contacts. This was initiated by the previous managing committee of Bombay College of Pharmacy Alumni Association, and we carried this tradition forward, where we invite our elite alumni for technical talk and an opportunity to share their achievements. This also opens up an avenue for our ex-students who have interaction with them and speak their interests and also seek some mentorship in their area of interest. Today, we have 2 very eminent persons with us who Dr. Mala Menon, our very own adjunct professor, has invited to give a talk. Dr. Anita Nair, who's joined us from Germany, and Dr. Suresh Kumar from India. Before I hand over back to Ashish to continue with the session, I would like to make a couple of announcements. We have received an appeal from the college once again this year for contribution towards the Students' Mutual Aid Fund. As due to the current COVID crisis, which continues, there are many students who have appealed for funding for continuing their education. We request you all to contribute once again wholeheartedly for this noble cause, either directly to the college, stating that it's an alumni contribution, or send us the contributions and we will collate and support the college. The second item is we are planning to release a newsletter from Bombay College of Pharmacy Alumni Association for the very first time on January 1st, 2022. Do watch out for the same and have a good reading. I would like to take this opportunity to wish everyone happy holidays and a wonderful new year ahead. Hope we all can meet in person next year and continue with our tradition of Chai Pe Charcha. So thank you very much, and over to you, Ashish.
Thank you. Your wishes that we should meet physically and have a next Chai Pe Charcha in the college as usual. I'm sure it will be coming through, and this Omicron will not be in the picture till that time. That's what we wish for. And thank you for reminding the SMAF, because today only Dr. Swati had shared with me per student what they are expecting, and also I'll share that, and we'll make an appeal to our alumnus also. Thank you very much, madam, for this initiative continuation and your address. I will now request Mala madam to come forth and introduce our first speaker. And I would suggest to all that let the speaker finish the session, and the questions can be put in the chat column, or they can be asked at the later after the speaker finishes the session. So I would request Dr. Mala to introduce our first speaker, so that we can request the speaker to put forth their thoughts. Over to you, ma'am.
I think, how would I share the slide? You'll have to make me presenter, or?
I think Agnivesh.
Yes, sir.
Is,
Yes, sir.
Make multiple participant as a sharing, because speakers also would like to share.
Yeah.
You are the host, sir, at present. Host is you.
Okay, I am a host. So done, ma'am.
Very good. Good evening.
Your slide is not visible, doctor.
No. I have not.
Okay.
I'll just thing. Shall I share now? Should I?
Yeah.
Yeah. Okay.
Whenever you wish to.
Yeah. Okay. I thought you're still this thing. Now is the slide visible? The screen visible?
Yes.
Everyone, okay. Now I'll just put it on full this thing mode. Yeah. Now it is clear, everyone. Yeah. Okay. Very good evening to one and all who have joined this Chai Pe Charcha session. Many thanks to Dr. Anita and Dr. Suresh for having accepted this invitation and give their speak about their journey. And it's a pleasure to introduce Dr. Anita Nair, who was our student at Bombay College of Pharmacy in the pharmaceutics section. Dr. Anita Nair is currently with Merck in Germany. She's a regulatory expert for API and excipients, pharma registration in life sciences, regulatory management. She is a BPharm from three very eminent institutes, BPharm from ICT, MPharm from BCP, and PhD from Goethe University, Frankfurt. After BCP, that is after completing MPharm, she initially joined academia. So right from 1999 to 2006, she was faculty in the Department of Pharmaceutics with MET and SVBCP in Mumbai. Subsequently, 2009 to 2013, she completed her PhD in pharmaceutics with Professor Dressman, who is very renowned. I think everyone knows about the Marcel Decker book by Dr. Dressman at the Institute of Pharmaceutical Technology. Now, then 2013 onwards, she has been with Merck in several positions. Earlier, she was head of Laboratory of Solubility and Physicochemical Properties Lab. And then she is now in the current position as a regulatory expert. In this position, she handles several responsibilities like preparation of dossiers for APIs and excipients, supporting life cycle of DMFs, providing support to LS business, especially with excipient registration in China, conducting training programs for internal sales team, customers, providing regulatory support to new product launches, and contributing to business strategy. Now, several other achievements of Anita. She has graduated talent programs, international managerial program, site management mentoring program. She is a beneficiary of pharmaceutical education and research with the regulatory links or PEARL, European Training Network, through Horizon 2020. She has a key account manager. She's a key account manager with cleaning validation support in biopharma and healthcare sector in Billerica, US. She's an invited speaker at several international pharmaceutical conferences on pharmaceutical technology, biopharmaceutics, and regulatory affairs. She has several scientific publications to her credit, mainly in the EJPS. She has edited and published a textbook on solubility in pharmaceutical chemistry with De Gruyter of Germany. So I think Dr. Anita is a very accomplished academician and also in industrial pharmacy. So today she will talk to all of us, which is mainly targeted towards the younger alumni and students from university to industry, how to be future oriented. So over to you, Anita. We look forward to listening to you.
Anita, you're on mute. Dr. Anita, you're on mute.
Yeah, now you can hear me?
Yeah.
So thank you, ma'am, for your introduction. Highly appreciated. And I thank all, especially Mala ma'am and Parizat ma'am, for the opportunity to come back as an alumnus, although virtually, and then talk to all of you. So what I have planned for today is just take my journey across, right from doing my B.Pharm days all the way till today, and just show you how within the whole process, there were things that went throughout my career and how it helped me to train myself or adapt myself to the future of the pharmaceutical industry and to keep us live and viable within that industry. So the talk, that's why it's named From University to Industry and How to Be Future Oriented. Just before I start, all the opinions that I'm going to tell here is my personal opinion. It should not be taken as something coming from a more policy or view. So it's just for all the audience to clarify that. So Mala ma'am already showed you my whole career journey of what it is. This is just an overview of the last 2 decades as to how I was evolving in my journey across the pharma field. So I started out pharmacy in UDCT and did my M.Pharm in BCP, wonderful periods. Then as a lecturer for biopharmaceutics and pharmacokinetics, I remember that biopharmaceutics and pharmacokinetics was introduced in M.Pharm as an elective, and Dr. Tipnis used to teach us, which had always, I had a strong liking for it, and pharmaceutics was always something I was attracted to. So I did my academic career in these subjects, moved to Germany for personal reasons with my family, and then had the opportunity to do PhD with Dr. Dressman, which was also a great experience for me. And since then, I have always been in Merck. So presently, I was the lab manager there for 6 years, moved as a regulatory expert. I will come to the details in my next slides. And then from next year onwards, I will be heading the global department. That's the regulatory management department for pharma food, which will be involved with the registration across the globe for APIs, excipients, and as well as advocacy and consultancy. So my talk will basically encompass my academic part of the career, the industry part of the career, and I will close off with a few key takeaways that I would like to give you all from my perspective what would be required if we need to move in any of the directions. So usually what I have observed within my networking group is people move usually from industry to academics. There are very few people who move from academics to industry, but then we have seen that the experiences that you get through academics, it really strengthens us or makes us prepared for the industrial, approach. So I will highlight on those as the talk goes by. So as you all know about drug discovery, it all starts from medicinal chemistry, target selection, compound discovery, goes into preclinical phase. At that preclinical phase is where pharmaceutics comes into play, where we start finding out or adapting what would be the best approach to formulate the products, and then goes into the clinical study, the marketing part. Of course, the regulatory comes into play quite in the earlier stages, and then you have the product in the market and the post-marketing phase. This is a very long period. So usually this is when I was a student of pharmacy, this whole period, we are talking about more than 10 to 12 years, the whole thing happening. Over the period of time, this has shrunk drastically. And I will come to that, how this period has become shorter over the period of time and how things have evolved in the world of pharmacy. As you can see, when you start from the early state, that's the target selection state, you come across a large number of molecules. And then finally, it's just that one single molecule, which could be a blockbuster, which comes out into the market. Yeah. In that period, when I was studying and doing my pharmacy, so at this time period, this, as you can see, what was happening then is the products in the market were more new chemical entities, which were chemical entities, and the biologicals were not that much. So those were an unknown area, or people were doing research, but products were not coming into the market. Usually highly soluble drugs were more flooding the market or coming through the pipeline of research, and not poorly soluble drugs. It was, it was more the normal kinds of drugs, which was easy to formulate. If a drug had poor solubility, it was thrown out, was not considered as a candidate. And that's how the trend was going. At that time, I remember that the BCS classification system was presented by Dr. Admedon. At that time, a paper was published. And I remember Dr. Padma mentioning that in one of the pharmaceutics lectures about this classification. I never realized then that I would do my PhD based on this concept. It was interesting. If you look now at that particular paper, it was bringing two basic concepts which we knew every time. We knew that when a drug has high solubility, it has a poor or a lower log P value, the permeability becomes an issue. So hence the solubility and the permeability has to be optimized. And here Dr. Amidon and his group, they came together, they picked up data, they evaluated the existing data and helped to classify products into highly soluble and poorly soluble, as well as with the permeability factor. And that opened a totally new aspect of how we looked at drugs and molecules, how pharmaceutical, people started looking at how do you formulate it, how do you devise it. Around the same time, then moving to when we were doing our masters, I remember Dr. Coutinho was involved with computational chemistry, where I had a few of my colleagues who were students with him, was using computational modeling approaches to design a drug. because the way we were understanding molecules was increasing. Our knowledge in biopharmaceutics was increasing. We were understanding how the drug behaves in the body. What does the body do to the drug? What does the drug do to the body? Similarly, advanced drug delivery systems were coming into play. There were talks about liposomes. I remember during my MPharm time, Dr. Vavia was stepping towards the, his students were working on using pre-liposomes for formulation techniques. It was quite a wonderful interaction to understand how a molecule or this lipid construct can take in water soluble and water insoluble or poorly soluble drugs in the same formulation and help that in drug transport, even targeted drug transport. So what was happening is drug discovery world was finding out new boundaries. At the same time, in Pfizer, Lipinski came up with the rule of 5. Of course, now people are cutting through that boundaries too. They are going further. They are no longer sticking to that. If it's a log, you have a high log P, I will not take this drug. No, there are drugs which are coming into the market, but it was changing the way we were looking at things. And these formed the foundation for us as students, because I will show you over the journey how these got implemented in the pharma world and how they led to the new pharmaceutical approach, even including the COVID vaccine that we are talking about, based on these things, how it was evolving in pharma. So at that time, I was teaching. And again, I can tell you, I got my first job because I was ready to teach biopharmaceutics and pharmacokinetics, which was introduced in the BPharm level. That's where I started at BCP. I was teaching pharmaceutical technology, dispensing pharmacy. And during this period, that is from my BPharm days till my teaching career, there were quite some skills that was being developed. And I thank all my teachers as well as, everyone, even my colleagues at that time. It helped me even develop the soft skills. How do I interact? I remember I used to take practicals with Mala ma'am, and it literally helped me to talk to students, understand them, explain things to them, improve presentation skills, how to write an article, because people take it trivially, oh, writing an article, just put things together. No, it's not that way. You need to have a skill. Your article has to tell a story, because now I review articles for EJPS, the other journals, and you have to see that there is a flow, and all this usually comes at this basic stage, and academics builds a lot of this. Similarly, networking, getting to know people across labs, not just within your team, but even across other, like we used to interact, what's happening in chemistry, what's happening in other colleges. Even now it's become much more easier because of technology, that you can know how research is happening in various cases. So these are the skills that was also developing simultaneously, and it really helped to understand the scientific know-how, the research skills, troubleshooting skills when we were doing our research part, and then, oh, we got stuck. So try to find solution or bounce the idea with your friends and try to see how the others think. Is there some aspect that you have missed? All these things were quite useful for me to develop and to bring that in the later stage of my career, which helped me to actually enhance the understanding in the science and the pharma field. Again, it helped me also to keep my knowledge up to date about new technologies, about concepts, reading articles. So this was really, I would say, a sponge period where I was getting so much information and so much of my, knowledge at that period of time. So the skill sets that I basically see, or I think are key at your academic phase, is one, a good way to get your scientific knowledge up to date. get your basics right. Science is huge. It's a ocean. We can't know everything, but whatever you know, get the basics right and keep your knowledge fresh. It keeps changing. Concepts keeps changing. There is a new dimension added to it. So you need to be constant touch and learning on what's new. Communication skill is something that is required across your career. If you are not able to communicate your knowledge, your concepts, your ideas, your strategies in a very simple manner that the audience can capture, you have lost them. So you need to be to the point, try to communicate, and this is very important. I feel in a way the teaching career that I picked up really helped me there, because today if I can communicate, get across a big group in a meeting, in a very simplified way, I usually then get their attention, and it creates a very big impact. And that's something that all of us should work deeply on. The research techniques, knowing new technology, analytical techniques, these are important. I remember when I was doing my master's, HPLC was a new technique. Today, HPLC is there in every monograph that you see. We used to do our evaluations using a UV spectrophotometer. Now, if you look at the monograph and compare it, they are going for liquid chromatography with MS or HPLC, which is all hyphenated systems. So it keeps evolving, and you got to be in touch with the newest technology and what's in. Networking is also key. So going to conferences, associations, interdepartment. So don't just confine yourself to your topic that, oh, I'm doing pharmaceutics, so I just need to know about pharmaceutics. No, you have to know the bigger picture. See what's happening in pharmacology, what's happening in chemistry. All this adds for you to understand what are the key things, what is important to bring things together. Going a bit ahead, then as years pass, so this is coming to the decade of 2000s. This was again the teaching period, and also around 2006 is when I moved to Germany, again for personal reasons. And this was around the time when Germany had hosted the World Cup. For the Football World Cup, and we came here, and believe me, nobody spoke English in Germany. It was all German. So it was a challenge, because even universities here, they teach pharmacy in German. What was happening in the pharma world at that time, again, if you see, the NCEs were dominating the pipelines, and they were coming out into the market. But at the same time, now the poorly soluble drugs were coming into play. And the reason being the understanding that we had on how to formulate a poorly soluble drug. So you remember I spoke about the BCS, the advanced technologies which were evolving in the earlier years. They started being used to push the drug into the market. So people or the industry started taking risks and calculated risk and pushing such kind of drug into the market. At the same time, the ANDAs, or the abbreviated or generic, to put it very lightly, the generic drugs, more were being pushed in the market, because if you look at the FDA statistics, the BCS concept by the FDA was implemented around 2000, 2001, because around 1995, 97 is when Dr. Vinod Shah and Dr. Amidon and group came together with the BCS concept. It was for the FDA. And then the biowaiver concept started developing. And then the generics were coming out. And around the beginning, there were only 3 or 4 which came up. But in the mid 2000 years, more and more generics were coming out with the BCS concept, where class 1 and class 3 drugs were being pushed out into the market. So this year shows only the new chemical entities. But as I said, those concepts which was being developed in the late 90s were being used to push the drugs in the market in the mid and the late 2000s. So what was evolving at this time? Again, the computational part of drug research did not just confine to chemistry now, it also came into drug prediction. PBPK models were getting more stronger. I remember around this time in Dr. Dressman's group, they were using the Stella model, which is a very basic model to put together the PBPK concepts. GastroPlus was coming up with their concepts, and they had brought in the more commercial form of the PBPK modeling. Similarly, SimSip was giving you a population-based prediction model. So what was happening is, in drug research, it was not more of a trial and error. It was not that just put things into animals, test them and pull them out. They were using animal studies, but at the same time using the understanding in biopharmaceutics, the computational model, to know exactly what is a potential molecule so that you reduce the risk of your product in the clinical phase, because the amount of money that is pumped to get a drug product in the market is really huge. We are talking about millions and millions. And at the same time, you got to be sure this molecule has potential. And these predictive models were very useful at that time. Around the same time, Professor Dressman came with the concept of biorelevant media. It was because, as you know, we moved from a highly soluble substance, which could be easily tested in aqueous buffers. From there, we moved to a more complex system like poorly soluble drugs, where we wanted to know how it will behave in vivo. And for that, we saw that we were using surfactants, but they were not exactly mimicking what's happening in the stomach environment, in the intestinal environment. And that's when the biorelevant media came into play. I have seen this media being prepared in the lab. It was a long process, a really long process, where I saw my colleagues literally using an evaporator to make this media, evaporate the solvents, get all the lipids inside. And that's when biorelevant came into play, and they started manufacturing it in a commercial way. At the same time, the biowaiver concept, which FDA brought in, started being accepted by other regulatory authorities, where the WHO came with their biowaiver concept. This was authored by Professor Dressman then, and then there was the EMA who did that, and Health Canada came into play. So because we were part of that research group, we were also involved, our team, in authoring and writing these concepts. So you saw that BCS that was introduced to us as a paper by Dr. Padma, it actually started, playing a role in the career path, because we were like, yes, I know this concept, and we started working on that together. The enhanced technologies, because poorly soluble drugs were being used, these enhanced technology like nanoparticles, co-crystal concepts, and these started coming together quite closely, and we started understanding, because they also had an influence on the solubility, the pharmacokinetic aspects, and how would a regulatory authority handle a co-crystal, because no co-crystal has come into the market as yet. So it is, it is a different way on how such particular molecules will be considered from a regulatory aspect. So during the research time in Germany, in Goethe, it was giving a holistic approach on how to look at a concept, not just with the idea of having a research paper. Rather, is this concept viable, which can go out into the market? How will the regulators look at it? How can you predict things about its performance? It was more of a holistic approach, what were being done there. So during my time as a research associate, I was involved with all these regulations, evaluation for the WHO, the guidances, even writing the comparator guidance for the WHO, at the same time giving talks in conferences. My biggest challenge during my PhD was not getting to know the research and stuff, which was already, the basics was being formed. It was, I was pushed into cold water where Professor Dressman asked me to take dispensing pharmacy practical for second semester students in German. So it wasn't that I didn't know the subject. I knew the subject. I have taught the subject in India, and the syllabus was the same. I didn't know the language, and I had to talk to the students in German. The reason I'm highlighting that here was, it was the biggest challenge that I had. I remember I used to be awake till 1 o'clock in the night and write on top of the words in Hindi how to pronounce it. I can give you an example. When I had to teach them the Noyes-Whitney equation, and I have to say solubility rate, the students never understood English. So, and solubility rate in German is called Lösungsgeschwindigkeit. So you know how different it is. It doesn't sound the same. So I had to learn this language in order to communicate my dispensing knowledge to the students. It was challenging, but I can show you later how it helped me, because I learned this language. It today helps me in my day-to-day work when in the position that I am in, to interact with people, because as I said, communication is the key. And if you don't know the language, I'm not saying all of you would end up in German, but there would be situations in your career where there will be something totally new. And in that case, pick it up, take that challenge, and then it will help you further, because it's a new learning. It will help you learn a new language, and you even learn to communicate with people more. So coming to that, the skill sets that was being developed at that phase of my career was collaboration. Here I saw that research in institutions were not just about doing it and keeping it within academia, but also extending it to the industry. So there was more industry academia collaborations, and I was seeing this trend also in India. Again, in my alma mater, be it in UDCT, be it in BCP, I was seeing that there were quite some interactions happening between industry and in the academia, and the research was very focused in bringing more and more skills and more new concepts within the industry which can be implemented. Digitalization was really becoming strong. All these computational models. Regulations were being a key, because even in the regulatory world, the aspects were changing, the approaches were changing. The regulators were becoming also a bit bolder and saying that if you can scientifically justify a certain thing, concepts, we are ready to consider them and accordingly adapt regulation. So there was, that conservative approach was slowly being left out. So these are the concepts that were coming into play, sort of like novel excipients, be it, or even ADCs now, which has come into the market. And last, what I would like to say here in this part is the calculated risk. So the reason I gave you the example of the German was, you should sometimes go out of your comfort zone. It's okay to go out of your comfort zone and try to risk it. See whether it works for you. Maybe you fail, but at least you will know that you tried it. You should also think outside the box. Don't follow what is always been the trend. It's okay to think outside the box and try it out. It may fail, but as Einstein say, one additional way how it won't work. So you can look at the remaining 99 ways. So always try to take calculated risk. Don't go over the board, but calculated risks are always good. So that was the academic part of my career. Then coming to the industry. So during the end of my PhD, that's where through network. So what happened is, my ex-boss was doing his habilitation in the same university with Professor Dressman. And that's where I met him. And I got an opportunity to join Merck as a lab head. So what was my task there? So I'm showing you the same picture again. So my lab was basically supporting the preclinical study phase, and I was supporting the medicinal chemist to do physicochemical analysis of all the new entities that were being done. So in a month, we used to get close to 100 to 200 new NCE molecules in a really tiny size, around 10 microliters of the molecule used to come as a DMSO solution in my lab. And we used to run close to 600 experiments, on a monthly basis. And then from this 10 microliter solution, the team had to upload close to 5 data. So we had to do the log P, log D, the kinetic solubility. These data had to be put into the system. And this had to be done fast because the chemist wanted the information within a matter of 1 week. And you know that this is just not possible with a lab of 5 people. And we had other tasks to do also, because we were supporting also the pharmaceutical scientists in finding the right formulation, doing solubility studies, doing the profiling of solubility. So naturally, we will use technology, and we had a robotic system within which we used to prepare plates for us. And these were then analyzed, and we used to then evaluate the chromatograms. Now that means we are talking about lots of HPLCs in the lab and everything. So understanding the HPLC concept in the M-Pharm days, and then using it as a full-fledged analytical tool in the lab was very useful. So we had to do troubleshooting with HPLCs, and sometimes you have to see, is the data correct? Maybe the data, because our chemists are relying only on our data. Let's say you use an ibuprofen and determine the log P. You have the data of ibuprofen published in a literature, and you can compare and see if you got the right results. But in case of this kind of concept, there is no physicochemical data available anywhere in literature. So whatever my lab was delivering was the basis on which the chemist used to look at their molecule and design it or redesign it in order to create another molecule which will fit to the bill. So that means they had to rely on the data that was being given. So the number of compounds that was screening was high, but the amount of compound that was available to be tested was really, really tiny. So 10 microliters is like nothing, but we had to give information with that particular amount, which means we had to devise analytical techniques that were really tiny for small quantities, very small quantities. We cannot say, give me 1 gram of sample. Like if you say something like this, that chemist will never talk to you again. They'll say, no, I don't want your lab. You are asking for too much. So their first batch used to be in milligram concentrations. And then we used to just get probably 1 or 2 milligrams out of it, with which we had to work. We had to do even the animal vehicle screening with such small quantities. So at the same time, at the pharmaceutics level or the pharmaceutical technology level, there was this challenge that we have prediction models, but a prediction model is as good as its input data. So we needed dissolution studies, but the dissolution studies, what we have in the market was for established drugs, for larger compounds like 100 milligram doses or 1 milligram, and we don't have the quantity. So as you can see, I've shown here some examples of how the mini dissolution was being developed in academia, even in industry, and how they were trying to mimic it very close to your gastrointestinal tract. So as you can see here, there was the AMI system, which was being used. This was in Denmark. The BioGit, which was used in the Greece University to understand the concept of transfer. Professor Dressman at that time was using the transfer model, but again, in the big dissolution apparatus. This was the TNO system was being developed in Pfizer to understand how drugs are absorbed, what is the pharmacokinetic data, so that they can put that in GastroPlus. This was in Greifswald University, where they were using GastroDuo to understand how the gastric emptying was happening. Similarly, they had the artificial stomach duodenum model. And then this was, again, Dr. Amidon working on the biphasic dissolution system. And we in Merck at the same time also had this challenge, because they were looking up to my lab asking that, how can you do something so that we get a transfer model? because I was in Professor Dressman's lab and had understood the transfer model well, because I was working very closely with the other team, we devised a very simple transfer model within our lab. So all it was we just took 2 conical flasks, put the gastric medium in one, the intestinal medium in the other, and we put a peristaltic pump in between, which was mimicking the gastric emptying. And we were trying to see how it behaves when the poorly soluble drug moves from stomach environment into the intestinal environment. And with that, the advantage was, if my drug had a dose of 100 milligram, I just needed around 10 milligrams to do this kind of study. And this amount of study at a preclinical level at such small concentration was really helping us to find out what is the right molecule that should go in the next stage. What did I learn from this? As we have always learned from our teachers, keep it simple. Sometimes you don't have to go for a complicated solution. If you can just put a simple model together, it will give you a lot of picture on how things are moving or how the drug is being evolved. And with this, I can tell you, we screened so many molecules at that early stage, and few of them really have hit the market. Yeah. So this was not a new concept. All we did is we made it a bit smaller. We fit it to a size which was useful to be used at a preclinical level. So these are the kind of approaches that people would expect from you in an industry if you are in an R&D level. It's not the only one. This is just an example on how you can do it. So what was my roles and responsibilities as a lab manager? So if industry is what you're looking for, if you are thinking about, in these concepts, here it was more on pre-formulation and physical characterization. So remember, your Martin book is very important. Many people, I have heard during my college days that people say, oh, I don't want to learn Martin. No, it's important. It's really important if you want to talk with your chemist. I had to also do technology scouting. So find out what are the new technologies in the market, try to see what fits in for our needs, for our research groups, so other research groups, of course, leading and managing key accounting, getting to know the digital tools, how to automate, how to do automation within your lab. It had just started when I started being a lab manager, and now it has increased over time. Keeping in touch with your basics again. So all the skill sets that I mentioned in academics are also relevant here. And the Pearl project was a wonderful project where we had a 3-way interaction. So it was a consortium of 5 regulatory groups, 5 academic groups, and 5 industry groups coming together to evolve in silico techniques, evolve formulation techniques, and also to make mini tools on which can be used in industry. So that was also a 4-year project, which was really, keeping us up to date with what is new within the industry. So here, what are the skill sets that I would present is collaboration. It's important to have an interdepartment collaboration. My ex-boss used to give me a very good example. If you want to make a house, and you make a house with, out of gold or of diamond, the best brick, you are making the brick. So you are responsible to make one brick, the other department has to make the other brick, and you made the most polished gold or a diamond brick and said, this is the best work I have done. It is still of no use, because the brick has to fit the house. And if the brick is not rough enough, you cannot put cement, and it will not serve purpose in the house. So what was he trying to communicate to me was, you have to do your work to the best of your ability, but you have to see the bigger picture, that I am making this brick which has to fit into the house. So the brick has to fulfill all the requirements required to build the house. So it's not just your brick what is important, it is the house is what is our target. So that is what was trying to be communicated. So hence it is important to understand, what does the other department want? What should I do? And adapt yourself and provide to the team and not just for yourself. So in silo working is no longer the industry trend. It is a teamwork which comes. So always see the bigger picture, digitalization, understand the business perspective. You need to be quick. You need to be fast. The one who comes to the market first gets the bigger part of the pie. So if you do your work gradually because you want to give the best work, it doesn't help. Time, efficiency, and quality all matter. And of course, if you're leading a team, leadership skills, people skills, you have to be empowering and you have to coach, because you will not be doing the test yourself. Somebody else will be doing for that. So there has to be a level of trust, and also you should have the ability to understand data. So these are the skill sets that you would require when you are doing that part of your career. Now coming to the last phase of my industry where I was as an expert. So that what I was doing was for the healthcare side. So if you know, Merck is having 3 major pillars or division. We have the healthcare, we have the life sciences business, and we have the electronic business. So in the electronic business, more on semiconductors, pigments, and that. Life sciences, we are more into APIs, excipients. So we supply these to the healthcare or the pharma and biotech industry. And the healthcare concentrates on new chemical entities, new molecules, that is more in the direction of oncology. I had a good idea on what was happening here. It gave me a good overview. But the difference between pharma and life sciences is, in pharma, as I mentioned, to move from this level to this level, the fastest that you can do that is close to now 3 to 6 years is what you can look at. But in life sciences, you cater to every part of this whole chain. So because I had a good understanding of what things were happening, it helped me settle here quite quickly because I exactly knew what was the requirements happening there. So as a regulatory expert role, it was helping me to provide the right suggestion to big pharma companies. So we have quite some big pharma companies who are our customers, in giving them the right regulatory advice, and also helping them out in choosing the right APIs or excipients quality when it comes to providing them that information. So what I presently do in my present job, it is preparation of the DMF files for APIs. So DMF are drug master files where we need to submit it directly to the health authority and the customer can refer it to. The life sciences business provided me a complete overview of the business. So it was showing me how the other pharma industry thinks. It was giving me an idea, how does the production department think? How does the procurement think? Because as a regulator, it's not that I just work with quality data. I have to work with the QC lab. I have to work with the production lab. And if you have to talk to them in Darmstadt, you need to know German. So now this is where the German skill was helping me to interact with my team and also with the people who are involved in giving us data. Because if you go to a production, a person who is actually involved in synthesis, in Germany, he speaks only German. He doesn't speak English. So you need to interact there. And there is where I thank Dr. Dressman in pushing me to learn German, because that helps me today and makes communication very, very easy. Not only that, it gives you exposure to global markets, the regulation, it gives you a holistic look. And I am involved right from the product launch or project conceptualization, to be more precise, to the launch of the product. So you know how is the whole life cycle of the product and what happens once it comes into the market. China regulation is now a big thing in the last 5 years in pharma world. China is a very critical market. Every pharma industry, Pfizer, Novartis, BioNTech, name it, they all want to go to China because it's a big pie. And for that, you need to know the regulation. And this is one country where the regulations change and are implemented in a short time span of close to 3 to 6 months. So quick is the change of regulation and implementation. And this is the only country in the world where excipients have to be registered. So this was a learning experience for me. At the same time, presently, I'm also involved in the two major vaccines which you know of, the BioNTech as well as the Moderna vaccine. So the excipients that are used in it come from Merck. So we are responsible for registering these dossiers and in providing support to BioNTech and Moderna for the COVID vaccine period. So it has to be quick because you know it's a pandemic. It needs to have the regulatory data for the product to come into the market. So the last 2 years has been really lively and at the same time satisfying to feel that, oh, I have contributed to the situation in the pandemic where I was involved in supporting something that is coming into the market, which could be saving people's lives. So this was a very good experience. And based on these basic knowledge and everything is how I will be taking up this new role next year, where I will be heading the complete global regulatory team within the life sciences for the registrations as well as the advocacy. So finally, what are the key takeaways that I would want to say before I conclude my talk? The skill sets that I have shown you so far, I find it personally, they are all important. You can't just say that, oh, that I used in academic, now I'm in industry, I don't need that. No, they are all important, but what one or the other will have more prominence or more priority depending on what you are doing at what time. So when it comes to academics, it's very important today, as of today, that communication skills, your scientific contributions, innovative thinking outside the box, and of course, your industry academic collaboration. These are the keys that I feel will actually drive you in making a career within the academic part. When it comes to industry, giving my R&D and regulatory kind of knowledge, the innovations, the application, how to put that into practice, networking. Again, when I say networking, I'm talking about networking within your your company as well as outside your company on these topics. Digitalization and artificial intelligence is the next big thing. So just don't, the new generation, you are very good at digitalization, smartphones and stuff. You learn it in a jiffy. Keep continuing that. This is what is going to happen in future. Now we are having retro computational chemistry coming up with artificial intelligence. They are working on that. And that is what is the new thing going on in the industry. Soft skills is something you will need completely across. Even if you are not thinking about making a career, you need these soft skills. Just keep improving them. Again, continuous learning, communication skills, team player, no in silo working doesn't work anymore. Taking calculated risk. And here, when I say networking, if you see networking is written twice, it's not that I have missed it out. It was done purposely. When you say networking, here it's more of scientific networking. When it comes to soft skills, sometimes you come across people who actually have certain soft skill abilities which are really impactful. Network with them, you learn a lot, because learning is something you should always continue. So always stay curious. And as Henry Ford says, anyone who keeps learning will always stay young, be it your career or be it your life. You have to learn for life. So as long as you learn, it will always keep you viable. Thank you for your attention. And that's all from my end. Thank you.
Thank you, Dr. Anita. It was a wonderful presentation. I would really appreciate your presentation skills, which you have not mentioned along with the soft skills, but it was definitely a very nice presentation for sure.
Thank you very
much. We could realize what you're going to speak, and we got every point from each aspect of your academics industry as other parts.
Thank you. Thank you for your kind words.
What you say, I fully agree that, yep, one has to, if you want to go ahead, domain knowledge has to be a pillar, no doubt about it. But as the technology, AI, digitalization, other things has to be mastered for going ahead. And more importantly, what you talked about, personal soft skills, that is 2 soft skills, you have mentioned all the 5 years, good. Communication and networking, these are the things which help not only for learning and sharing right away, but in the future, like when you go across and above 50, then you need those skills, because then people are there to implement what your great thoughts are. Absolutely. Excellent presentation. Thanks a lot for this lovely sharing your experience throughout, and it looks like a very big spectrum of achievements or contributions. What I see, luckily when you come as a speaker, we also realize, yes, we have good gems along with us. Thanks a lot for that.
Thank you for your kind words.
Yeah. Before we go ahead, there was a question. So yeah, if you don't mind, Chirag Dara has asked, ma'am, can you explain some info about the formulation development of anti-COVID pill molnupiravir and its efficacy on Omicron variant?
Okay, unfortunately, that's not one of my expertise. But again, with regards to that particular pill, I would say, I mean, when it comes to the pharmaceutical technology part and stuff, I think this was one of the drug ingredient which was under play or under test for quite some period of time. So that's the reason they could get it into testing and stuff faster. But again, as I mentioned, I'm not an expert, or I'm not involved with that particular topic directly. So that's why, unfortunately, I can't give you much of an information on that. Sorry about that. I can't hear you.
Can you hear me now?
Yeah, now it's better.
I'm taking a liberty of having a cup of tea because we are having
of course, it's chai pe charcha. Yes, absolutely. It should literally have that.
No, we do have physical meetings. We definitely have chai pe charcha. But I was more expecting, honestly, from you also, is that how Germany also has a very big scope for pharma guys to come and make their thing.
Absolutely.
Yeah, so that's what I lost in the whole.
So the point was this. So I can just tell you my experience, because when I started off, or as I said, I came to Germany for personal reasons. So my husband moved here, so we moved with family. So I should say the first 2 years when my son was just about a year old, I lost pharmacy because it was just looking after the kids. And then that's when I saw that Professor Dressman was heading pharmaceutical technology, and I just went to meet her for the PhD position, which she offered immediately. But there's one more thing that I would like to tell you, that at that time, which was very surprising, they didn't even know about Mumbai University. And, so although I had a master's and a teaching experience, they were still didn't give me a PhD position directly. They gave me a provisional PhD position, saying that I will have to attend, the 7th semester pharmacy classes, because somehow they said that our syllabus and their syllabus don't match. It was just because they never knew about our industry, because at that time, if you remember, there was a more flux of students to US and not to European countries. Lately, in the last 7-8 years, I've seen more students are coming to Germany and other European countries to do the pharmacy. So it can happen, this is just an information to the students, it can happen that they may ask you to take an oral test to prove your ability in research. And again, they don't ask you to, so they asked me to give an oral examination in a subject which is not my research subject. So I took pharmaceutical technology and I had to give an oral exam in German in pharmacology. So attend some seminars and then give it in pharmacology. Of course, they were kind enough that I could answer it in English. So you may get a setback there saying that, oh, they are not accepting. But as I said, take that risk, just come in, because Germany has one of, I would say, best universities and I was studying in Riedberg and near the Max Planck Institute, where there were also a few Nobel laureates who were having their labs. And you get a lot to learn. You have a good possibility. And there is a site called DAAD, DAAD.de. I would recommend the students to go through it. Many of the Indian and even international students get scholarship to come and learn within Germany in the various universities. And the pharmaceutical group are so well knitted, you can get quite some really good exposure here. One thing you should remember is, till now, there were no master's degree. So you do your B.Pharm. So it's not a bachelor degree either. In Germany, pharmacy is more of a technical education. So similar to, you become a pharmacist so that you can run your own pharmacy or take a job in a pharmacy. So it is seen very similar to medical education. And after that, you can directly do your PhD. So there is no M.Pharm, in between in Germany. Now there are a few universities like Heidelberg University where they have put in masters within their studying curriculum. So it can happen that you do your masters and come here, and you see that your colleagues or the other students in Germany directly after their studies move into PhD. But I would say Indian students have an added advantage because of the syllabus that we have. The technology part which we have is really strong, and that gives us one step higher advantage in compared to the students here. And of course, the language. They are very good at English, the Indian students, whereas German students, for them it's new because they have studied everything in German. So writing papers in English and all is a challenge for them. So this is the differences that you have. But yes, there is quite some possibility and quite some good universities that you can learn pharmacy in Germany. I can't hear you. You're on mute.
No, I was on mute. So thanks, I said. Thanks. And really, the one information which you gave was good, that Germany encourages people to come and do the technical education. Not only that, they help you fund education too. That's what you shared. So we'll definitely tell our students that if they want any help from Germany, we'll certainly get in touch with you. And if they want to get mentorship, they can connect with you also for German education planning. Thanks a lot, Dr. Anita.
Just one additional information. Every university in Germany has something called an international association. These are the group of students from different parts of the world. And they are a very good source of information to get the newest information. And also, if you get an admission and you land in Germany, if you are in chat with them, they even come to the airport to pick you up and to settle you down. So they have a very good association with the students. So these are some small things, because I know children are generally nervous that, oh, I'm in a new land, no language, what to do. But it's quite easy. So, but of course you can reach out to me and then I can guide.
Thanks. We would like to chat more, but the time is a constraint. And we started late.
I think Suresh should now jump in with his experience.
Suresh will have to. He was quite attentive. I could see every point he could also pick up, and he was picking up. I saw him. Agnivesh, are you there? So, Dr. Suresh, welcome him and to share him his achievements and what he's going to reflect on his career journey. I would request Dr. Agnivesh to introduce our second speaker.
Yeah, thank you, Ashish sir. Hello, Suresh sir.
You are audible. You are audible.
Hi, Agnivesh.
Yeah. Hello, sir. It's my pleasure to introduce you. It's my pleasure to introduce you and Dr. Suresh Kumar completed his M.Pharm in 2002 and PhD in 2008 from Bombay College of Pharmacy. He published multiple research articles and meeting abstracts. He worked with leading organizations like Glenmark, and currently he's working with Johnson & Johnson. He has over 10 years of experience in general toxicology, regulatory toxicology, and product safety. And along with that, more than 3 years of experience in pharmacological screening of NCEs, drugs, and natural products. He has expertise in risk assessment analysis for safety substantiation, as well as preparing and reviewing non-clinical sections of technical documents for regulatory submissions. He has excellent organization, communication, and leadership skills with proven ability in managing, coaching, and developing staff members to meet both corporate and long-term career objectives. He has received various awards for his outstanding performance in Johnson & Johnson. I know him since my M.Pharm days. At that time, he was doing his PhD, and he was in the completion phase of his PhD. And I remember him as one of the most respected senior who always mentoring and helping other seniors and juniors. Thank you, sir, for accepting the invitation for the talk. And I request you to please proceed with your presentation.
Thank you, Agnivesh, for the nice introduction. In the interest of time, I will just jump into the meeting. Can everyone see my screen?
Yes, we can see.
Okay. Is it proper now? Full screen?
Yeah, it's a full screen now.
Okay, fantastic. Good afternoon, everyone. I know it's we are at the end of the time, but I need Dr.
Don't constrict your information, please, for want of time.
Okay.
We would like to hear more. Yeah.
Thank you, Mr. Ashish. Right. Dr. Anita shared various very informative session it is, and a lot of information with respect to preclinical, pre-formulation aspects and other things. I would continue more specifically on the preclinical aspects. The preclinical aspects, what she described, is more towards the pre-formulation or formulation related preclinical aspects, while a subsection of the preclinical aspects are more towards the toxicology studies. So again, when I'm talking about toxicology study, it is very sensitive area for Johnson & Johnson, as well as various corporates as well, because use of animal, use of studies and cruelty, etc. are very well known public issues, and there are a lot of trend in talking about, this organization is doing this studies, that studies and other things. So any of the opinion which is expressed here is my personal opinion, not reflective of Johnson & Johnson as well as any regulatory agency. So this is about my journey. I just want to touch upon, where I started my graduation and then how I progressed through my career so far in one slide. I have to say thanks to all my teachers in BCP who have groomed me to this extent. Thank you, everyone. I see here today Mala Madam and Nagasankar Madam, Krishna sir, and I do not know who else have joined this meeting. However, thank you everyone for supporting and grooming. Your teaching, mentoring, questioning helped me to groom to this stage. Thank you very much. At the same time, I would like to acknowledge two important mentors in my life who have groomed me as a pharmacologist. One is Dr. Saraf, who is my supervisor as well. He empowered me in the lab to set up the respiratory pharmacology instrumentations. At the time, my primary work was on respiratory pharmacology. Of course, typical Indian research work, like I also picked up some herbal ingredient, worked on the herbal ingredient, but he allowed me to go to different institutes, including industry, to learn about how they are performing respiratory pharmacology experiments. What are all those new models? How can we bring those models to BCP? That was the thing. And that encouragement, that motivation allowed me to visit even Glenmark at the time when I was a student in BCP. I went to Delhi College of Pharmacy to learn about instrumentations and came back. And here, additionally, Dr. Vadlamudi Rao, the first director of BCP, he has a lot of faith in me at the time. He allowed me and he encouraged me to come up with various ideas of how to set up instrumentation. One is like you learn the techniques from outside and applying and bringing that on the table at benchtop is something really tough, because you go and visit industry, it is 1 day visit or 2 day visit. You go and visit other institute, it is 1 day, 2 day visit. But when you come back and then see here, you need really a person who can work along with me and guide me rather, and bring something value to the institute. That's where the experience of Dr. Vadlamudi Rao as an industry person helped me a lot. And I have to really thank him in terms of the setting up the lab, as well as the technical interactions in the pharmacology that has helped me to groom as a good pharmacologist at the time. As I completed my PhD, got into Glenmark, I was lucky enough, not every graduates get an opportunity to get into a job where they can continue the same research work or similar kind of research work. So when I got into Glenmark, I was in drug discovery area. I was working there for almost 2.5 years, screening various NCEs, performed various experimental models for asthma, inflammation, pain, allergy, all those models. At the same time, I had an opportunity to develop few models in Glenmark. Not necessarily this is totally a new model, but in Glenmark, I was establishing 3 new models. One for quantification of methylene blue-based assay for inflammation, and one whole body plethysmograph respiratory experiment in anesthetized unconscious animal. And we also purchased at the time one Bucksco instrument for measuring whole body plethysmograph in living animals. So again, in those 3 setups, I established models there. Of course, there are a lot of learning there as well. When you are establishing certain models, the opinions of me need to be matching with the lab supervisors. Another thing, there were difference of opinion. Another thing, how we convince, that's a key learning. How do you explain them? How do you take the lead? In my opinion, every one of you is a leader. There is nothing like leadership skill. Everyone is leader. Everyone has to have leadership skill. There are 2 levels of leadership skill. One is individual leader. Another one is people leader. How you handle people when you have people reporting to you, kind of thing. At individual level, you are the owner of your own activities, responsibilities, another thing. You have to plan, you have to decide, you have to think the end in mind and take every decisions and steps so as to accomplish everything in the right manner. That's what I did. I performed those experiments and showed the results. Yes, we can get the results by performing in this manner. And my supervisor eventually have to accept, because it is evidence. At the end, it is science which speaks, right? So successfully, I was doing those things. At the same time, I got opportunity to move to Johnson & Johnson in 2009. I would say it is kind of an accident. I came for the interview, and just in the interview panel, people from US, they came here and then they were interviewing, and I was responding. I did not know till the time I'm not going to perform any animal experimentation. So far, all the years, right from 1999 till 2009, almost 10 years, I was handling so many animal experimentation. Even as a part of industry institute collaboration in BCP, I was performing animal experimentation. I moved to Glenmark, there also performed a lot of animal experimentation. But when I went for interview, there was various toxicology related questions. I was able to handle those things, respond. Till the time I was a pharmacologist, but the interview was for toxicology position. Thanks to those interactions that happened at BCP, which groomed me, as I said, BCP taught me how to think. And those are all the things. So I was able to handle the toxicology related questions which came up during interview. Terminologies no one may be knowing, that is different, but you be logical, you be very clear in your communication, you be very scientific in your approach, systematic in your approach. You can clear any interview. You don't have to have prior experience of in for the particular role. That is not the criteria. One of the question after the question, everything was discussed. I would say, I asked them, "Do you have vivarium?" They said no. And I said, "I am interested only in a place where I would do some experiments," and I moved away. Probably they did not hear me well. Whatsoever reason is, I was hired there, and while moving, I thought, okay, let me explore this as an opportunity. And Johnson & Johnson, it's operating in 3 sectors: pharma, consumer, and medical device. I was recruited into consumer sector. I'm still in consumer health sector. It's the healthcare industry, and I started supporting OTC business requirements in North America in early 2009, and gradually I got expansion in the role for various regions. From North America, then the role got expanded to Asia Pacific, then Europe, then Latin America. So my current role is global toxicology lead for self-care business within J&J Consumer. We call this as self-care business. So I am the lead for OTC business activities. Before transitioning into full-fledged global lead for toxicology in self-care, I got an opportunity to work in oral care in parallel with OTC in skin health or skincare products, briefly for engineered products like sanitary napkins or band-aids, those type of products. And also recently, since last two and a half years, I started supporting dietary supplements as well, in addition to OTC. And all this activity as a toxicology lead, everything I am doing without any animal experimentation. So earlier I was a totally wet lab scientist. Now everything is based on computer, look into data, look into in silico predictions in some cases, and perform the toxicology risk assessments. I understood from the flyer and the prior discussion with Mala Madam, there are 3rd-year students, 4th-year students, and M Pharmacy students who are the audience in addition to alumni matter. So I wanted to touch upon a bit of basics of toxicology and a case study about risk assessment, how we perform, to give a real world scenario, how the risk assessments are conducted. And that's how I planned my session. So as a toxicologist, the important piece is to support the product safety or the material safety. Within the current context of discussion, we talk everything with respect to product So I restrict everything with respect to product safety. So we need to understand one basic fact of toxicology is the dose determines the toxicity, right? Which is very important thing. A small quantity can be well tolerated, while a large quantity can be very hazardous. Even salt, table salt, which is very commonly taken as food material, can be hazardous if it is consumed in excess, maybe 20 gram, 30 gram. If somebody consumes, they may even lose life. That's the situation for even table salt, which is very well-known safe component. Right? So when it comes to product safety, what I do is like I look into the ingredients. When I say ingredients, ingredients include the API, excipients, all those ingredients. And also, when I say API excipients, many a time there are pharmacopoeial grade materials, and for some of the products there can be non-pharmacopoeial material. Nonetheless, whether it is pharmacopoeial material or non-pharmacopoeial material, because of the synthetic process which was involved by the manufacturer of the material, there can be impurities which is outside the pharmacopoeia specifications. So this we have noticed, and we made sure that all those impurities also needs to be toxicologically assessed in order for the product safety, in order to safeguard the consumers and patients that uses the product. So, and when we talk about ingredient safety, APA safety, and other things, there are again two types of things, whether it is novel ingredient, be it APA or be it excipient. The requirement remains more or less same in the context, from toxicology context. In the context, we need to give whole battery of information, right from acute, repeat, and plenty of other informations, which I will just quickly cover in the next slide. When we have the ingredient which has a prior use already, like we know paracetamol. Paracetamol is already known thing, known pain, anti-pain agent. In these cases, we look into the paracetamol. Paracetamol as a material, it has been very well accepted for its safety. So we don't have to look at the safety of paracetamol. It is well established already. But the paracetamol synthesis may have certain impurities like PAP and other things. PAP is para-aminophenol. I'm just taking one quick example here. So what is the level of para-aminophenol present in the material? As per product usage, is there any risk to the patient when the material is used? That is what we do a risk assessment. PAP, when I say PAP has already information available, in those cases, we look into the data and then take those data and perform the risk assessment calculations. If data is not available, then there can be a need for alternate approach. We'll discuss that later. And as a part of product development, I also provide support for completing the non-clinical section of regulatory requirements, like in case of IND filing thing, we prepare CTD. So I provide non-clinical dossier, and even to conduct clinical studies in some cases for IB investigator brochures and CTA clinical trial applications. For all those things, the non-clinical section, which ensures the safety of substance or the material or the product, is necessary, because we all, most of us must be already aware, before any substance touches a human being, there is a preclinical clearance needed. Only when preclinical clearance is done, meaning like when it is evaluated and considered as safe, then only it can touch or it can be allowed for human exposure. Otherwise, regulators are not going to consider your application for conducting anything. We need to justify the safety of material, and that justification has to go from this end, toxicologist end. So that's why these regulatory requirements need to be fulfilled. And when I said I'm in consumer health, so it's not only, although my role is majorly on OTC, I have briefly worked in cosmetics area as well, where several countries, including India, has banned on conducting animal studies on cosmetics. I do not know how many of the colleagues who are attending this call are aware that animal studies cannot be, should not be conducted for cosmetics in India. So that awareness is very important, and what it led to, we will see in the coming slides. Because of ban on animal studies, there are, of course, alternate methods of toxicology evaluation need to be adapted, without which we cannot fulfill the preclinical safety requirement. On one hand, there is always continuous innovation. If we are not innovative, if there is no innovative products, ingredients, materials are identified to meet the requirement of unmet needs, the marketing things will be slower, because there will be always a competitiveness, right? Many companies may be producing these things. Most of the bigger companies, they focus on innovation, how to bring innovative molecules to address unmet needs. So we provide design studies for using alternate methods to address the preclinical studies. I will cover up that in the coming slide. And also, it is not only the new product, but also for the existing product. There are products which are already in market. Sometimes because of different storage conditions and other thing, we may find some impurities which are identified in the product, in those products. So impurity qualification, how do we approach those impurity qualifications? And I think that we can see briefly. And HHE is human health hazard evaluation. So sometimes because of impurity identified in the product from the market kind of thing, then we look up and then see whether are there any concern due to the impurity present in that. One is, because of scientific advancements, 30 years before we were doing UV spectrophotometry, at least some 15-20 years before the MS came into the analytical tool with the mass spectrophotometry came into existence, and people started measuring even small quantities. Nowadays, there are techniques like ICP-MS, atomic absorption spectrophotometry. All those things lead to even identifying levels at ppb level. So when such level of impurities are identified, it would trigger any identification. When it is identified, that would trigger investigation. That investigation need to be addressed and closed properly. That's why we perform that health hazard evaluations. Besides that, even for the existing product which are in the market, health agencies, they ask clarification question with respect to how did we qualify certain impurities in those products, how did, even for the active itself, there can be questions with respect to what is the safety of, say for example, pseudoephedrine in pregnancy and lactation, those kind of things. They ask some clarification question based on the labeling information, or SPC, summary of product characteristics, they call it in some region, especially in Europe. So those information, when it is available, regulators look into that and then they ask some clarifying question as to how do we mitigate those risks to the patients and consumers. So in those cases, we look into the database and then address the safety of those products. So when I say toxicology aspects, we look into, there are several toxicological parameters, and I listed something which is very commonly used. There can be few more which are not listed here. The list is not comprehensive, but it is certainly covering most of the things. So we look into acute toxicity information like LD50 data and other things, and we also look into the repeat dose toxicity information to mitigate the risk that comes from repeated exposure. What happens to a product if the product is used for 14 days or 30 days or lifelong kind of thing. In those cases, what can be the approach? And for at least in the area where I am primarily working is OTC. Most of the OTC are as per the regulatory classification, it is generally recognized as safe and effective. So the safety is mostly addressed on the active itself, but the impurity aspects are important. So when there is an impurity identified, whether that impurity is genotoxicant or mutagenic or not, all those things are looked into, whether it can damage the DNA or not, those effects. And if the product is used for long-term purposes, are there any risk due to the product, whether it can cause any carcinogenicity? Most of the all of the genotoxicants are known carcinogenic, but carcinogenicity can also happen through non-genotoxic mechanism, which is a threshold-based mechanism. So in those cases, it is pretty straightforward. Like we look into the information, what is the threshold available. In case of genotoxicity, you do not have yet defined the threshold. But in case of non-genotoxic mechanism, we know certain descriptors like no L, low L, no observed adverse effect level, low observed adverse effect level, all those things, and use those descriptor and derive what is the safe exposure limit for any impurity kind of thing that we look into. Of course, another important thing is the effect of any chemical to the reproductive system, especially reproductive development, especially for the pregnant women or mothers or parents who would be in the family way. Can there be any impact of the chemical or impurity or API on this system? Those information is looked from these reproductive developmental toxicity studies, and there are various studies, and that information is again considered. So various parameters of toxicity is looked into before we judge whether it is safe to the exposure or not. And of course, toxicokinetic is another important parameter. The reason is, if we know very well that the material is not going to be absorbed, for example, high molecular weight polymers or some metallic compounds whose absorption is very low, then by way of oral ingestion, the risk is relatively low. And especially if the route of exposure is dermal, these materials like high molecular weight polymers or the metallic compounds like zinc oxide and all, they are going to just sit on the skin. They are not going to penetrate through the skin. Unless otherwise they reach into the systemic circulation, they are not going to produce any toxicity. So that way, that piece is important. Besides that, how body handles those chemicals, what is the metabolite, whether the metabolite has any impact or not, all those aspects also need to be looked into in addition to the clearance, which is another important parameter from safety perspective. And the top things above the square box is all meant for systemic toxicity considerations. These parameters are considered for drugs, chemicals which have both systemic as well as topical route of exposure, because we know now that certain chemicals may penetrate to the skin. If they have penetrated, then they are prone for systemic exposure, and so the other toxicological aspects are important, right? On the other hand, the one which are in the box, dermal irritation, ocular irritation, dermal sensitization, photosensitization, these are other parameters which are certainly looked into for all topical products, primarily for skin contact product. This is very important for a toxicologist to determine. From the previous slide, if you see, there are a whole battery of information which toxicologists look into. Even I look into. You take any dossier or you take any complete profile of material, chemical material, you will find information related to acute toxicity. Even in repeat dose toxicity, you may find 28-day, 14-day, 90-day, 6 months, 1 year, 2 year, all those information. And again, with other parameters like carcinogenicity, genotoxicity, or reproductive developmental other effects as well. So when we look into the data, the primary objective is to see what happens to it. Like, is there any effect happens to the organs or systems in those studies? Then what is the dose response with respect to what dose that is happening? Is it dose dependent effect? That is very, very important. With gradual increase in the dose, are you seeing any increase in effect? Those aspects are very, very important for toxicologists, even for me. So I look into all these things, and various studies will give various information, right? Sometime within the same study, different organs may get impact at different dose levels. Sometimes different information may come from different studies, and they all point to different organ systems, liver, kidney, brain, or heart, anything. So based on that, the dose response is plotted, or at least the toxicologists interpret this information with respect to it and identify which is critical. When that critical dose is important, because critical is the one which at a lower dose produces adverse effect to the organ or system. So here in this graph here, you would see liver effect as a critical effect. Then we need to see what doses are not producing any effect. That is identified as no observed adverse effect level. What is the dose which is producing minimum adverse effect level? That is known as low observed adverse effect level. So based on that, we pick up the dose which is safe for exposure. That is NOEL in majority of the cases. At times, if NOEL is not established, it is also possible, because we do not know how the biological system is going to behave. When we finish the study and then we end up everything, and then later we realize all doses have produced some effect. It can happen. So in those cases, we need to determine what severity of the effect. Is there low observed adverse effect or low effect level? All those things need to be determined. And based on that, a right dose for the human studies and human exposures can be determined. So the primary aspect of risk assessment is to ensure whether the exposure is safe or not, right? So in the previous slide, we have seen how the point of departure, that are the descriptor, NOAL, everything is to be selected and identified. Once it is identified, then we have to calculate the margin of safety to understand what margin it gives. And this margin of safety is calculated, by dividing the NOAL with systemic exposure. The systemic exposure comes from what dose is administered to the human subjects and how much that translates to. Say, for example, if 60 milligrams is administered to a human subject of adult whose weight is 60 kg, then it becomes 1 milligram per kilogram. If NOAL is 100 milligram per kilogram and you have 1 milligram per kilogram, then you get margin of safety which is 100, and that 100 is minimum generally accepted as safety ratio by health agencies. While I'm saying this is a general acceptance criteria. There can be deviations. Those deviations needed to be well justified based on scientific evidences, based on ADM principles. Sometimes you may end up calculating PDs, permissible daily exposure, or reference dose instead of calculating margin of safety. Even in those cases, the calculation is done using NOAL with NOAL divided by uncertainty factors. These uncertainty factors are typically coming because of the NOAL is derived from animal studies. There is interspecies variation, animal to human variation, and there is intraspecies variation between human. You and I need not respond to a chemical in the same manner. To account that another factor, which is, that's why in the earlier case the ratio needed 100. That 100 also coming from this factor. Every uncertainty factor maximum factor is 10. So 10 multiplied by 10 gives 100. Even for deriving R of D or PD, there are different factors applied here. 10 multiplied by 10, and for the duration, if your exposure is study exposure is a shorter duration, while the intended exposure of the product is a longer duration, then we need to account the duration difference. So there should be a factor accounted. There are different factors given in different guidances, so those things can be adapted. And another important factor in the risk assessment is product specific risk assessment. Sometimes the product is topical product, skin product, cream, lotions, kind of thing. Sometimes the product is ingestible, like syrups, tablets, capsules, those type of thing. And there are different other types of products as well in the market, like we know that aerosols and sunscreen products, those type of products. So in those cases, we need to consider those special criteria. If there is aerosol, is there a potential for inhalational risk? And if there is a potential for inhalation, then the risk needed to be mitigated for the inhalational toxicity. So in those cases, we need to have the toxicity data from inhalational study itself. When I'm saying this, sometimes there are aerosols which are in the product, which are cosmetic products which are in the market. They need not require this because the particle delivered are larger in size and they would not get into the deeper lung. So they may not require an inhalation assessment. While in the drug industry, there are some products which are specific for deeper lung delivery. In those cases, we need to have certainly the toxicity studies conducted by inhalation route, and then the risk needed to be mitigated in that route. On the other hand, like sunscreen products, we need to certainly take into account the effect of UV lights on the product, sometimes by way of looking into the phototoxicity aspects, photosensitization aspects. Yeah, this is very interesting, at least for the cosmetic industry, and I am part of that. So I would like to bring attention and why this drove different perspectives in the coming slide as well. REACH is a European regulation. Anita would be knowing this far better than me. Over the years, they have decreased the or tightened the requirement. As you see in the graph, in 2010, for materials, meaning the substances, chemical substances whose produce is more than 1,000 ton per year, you need to submit all the information to the REACH as per the regulation. REACH is registration. I'm sorry, I not recall the complete thing. It is for registration and restriction of chemicals in European region. So it is a regulation. We need to make sure the informations are provided to the committee here by way of submissions, which are submitted in the ECA database, European Chemical Agency database. So every company who are producing chemicals in that tonnage, they have to give the safety data package, including the toxicological information and other thing they have to give. And substances which are classified as CMR, which are CMR means carcinogenic, mutagenic, and reproductive developmental toxicity substances, for which even if the produce was 1 ton or around 1 ton, they have or exceed 1 ton, they need to give entire safety data package. And over the period, the regulations got tightened. I have information here up to 2018. I don't have very recent information. I believe the regulation still remains in the same manner as it is. The final substance registration date, this one is more than 1 gram. Anyone who is producing more than 1 gram, they have to give all the safety data package to the regulatory agency. So that's how the regulations have tightened over the period of year, which means all the companies who are in the business, when the chemical is used in 1 ton in a year, it is not produced 1 ton, it is used 1 ton, I believe. If it is used 1 ton in a year, then they need to give all the safety package. Many companies, they form a consortium, they collectively write the submission package and submit to ECA in this process. While I'm saying this is one hand, they are asking for more and more information about safety of the chemical. On the other hand, SCCS, which is Scientific Committee for Consumer Safety, again, it's a European health agency which prohibited, this is the first agency in the globe which prohibited the use of conducting animal studies for cosmetic purposes. The first ban came in 2009 for the products. Later, till 2013, they allowed cosmetic, till 11th March 2013, they allowed the cosmetic ingredients can be tested. Post 11th March 2013, there was no study allowed even on the cosmetic ingredients. So there are several studies conducted prior to 2013, and they got the results, and those results were used for mitigating these safety aspects. Because there is a ban implemented, many companies which are in the business of cosmetics, they don't want to perform, or they cannot perform. It's not about want. They cannot perform any animal studies on cosmetic ingredients or products. So that's the big challenge that this industry, cosmetic industry, faces because of ban on animals. And one hand, there is always a need for more and more stringent data. The health agencies always ask for more evidences to say it is safe. On the other hand, another health agencies say that you cannot have any animal studies conducted. So how do we approach that? That's a balancing act. So that's where the alternate methodologies come into picture. There are various validated alternate methodologies available, which I have captured here for conducting toxicology studies. So there are in vitro, again, studies which can be conducted for skin irritation, eye irritation, oral irritation, skin sensitization. Irritation studies, there are studies for absorption as well. So using CACO2 cell line or MBCK cell line, studies can be conducted for oral absorption, and for dermal absorption, there are France chamber, diffusion chamber assessment assays are conducted. I know I was part of some of the experiments which had happened in BCP then. So, but there are a lot of other learnings when we actually perform at the industry level for the dermal absorption using the France chamber. So there are well-laid guidances available and recommendations available with respect to what is to be considered for identifying or establishing what is the dermal penetration of the materials. Those things can be referred through various guidances. The intent of this presentation is only to give top-level orientation on what are all different things that are being looked at. Similarly for genotoxicity, there are in vitro aims assay using Salmonella typhimurium strains and Escherichia coli strains. Different strains are used to perform this assay. And besides that, mammalian chromosomal aberration test, mammalian micronucleus test from bone marrow cells. These are different assays which are done to make sure that the material is not genotoxic. And regulators or health agencies do consider, at least on the genotoxicity front, a complete in vitro assessments are totally accepted. Likewise, irritation assays and sensitization assays are accepted by regulators, and absorption studies are accepted by regulations. We don't have alternate methodologies for some other things like repeated dose toxicity or for reproductive developmental toxicity. Although there is a ban which came in 2013, there are various organizations, private labs, and companies. They are working on alternate methodologies for these endpoints, repeated dose endpoints, and reproductive development toxicity endpoints. But nothing is, as of now, a validated model to my best of my knowledge. While whatever listed here are validated, including cytotoxicity using MTT assay. And one important thing which I would like to just cover up is the in silico prediction tools using quantitative structure activity relationship. There are different tools which are available, which I have captured here, like Toxtree, QSAR, Derek, Multicase, Leads Group. There are different, these all are validated. Some are freeware, some are you have to pay for its use, or you need to purchase the software for use, those type of things. So what is important is, like, certainly Toxtree, QSAR tool, these two are free. Likewise, EpiSuite is free. Finite skin permeation calculator, this is freeware. All these things are freeware. Anyone who is interested, they can just type these things, search, and then use. If anyone needs any guidance kind of thing in addition to how they can always feel free to connect with me anytime. of course, during daytime I will be busy, but we can just fix some schedule and then discuss that. What is important, another thing is that genotoxicity endpoint using the QSIR model has been well characterized, well accepted. When we say in silico model, this in silico model like direct, multi-case, lead scope, they have potential even to predict reproductive developmental toxicity endpoints. Those predictions are only at predictive level, but regulators do not consider that as acceptable criteria. So at the end, there may be a need for reproductive developmental toxicity for drug candidates. But when we use these models, multi-case lead scope or direct, SARA, these type of models, the QSIR, if it is clearly saying negative for genotoxicity, that is considered as valid and accepted by US FDA, TGA, and European Medical Agency everywhere. So I have personally different experiences with the different agencies. We have very much defended the safety of impurities using these models. And yeah, that's about alternate toxicology methodologies. I just want to give, because we talked about QSIR in the previous slide, what does it look like? How do we do kind of thing? So here is one case which I just wanted to quickly run through to give a real case scenario. Sodium lauryl acetate is one of the ingredient which is used in hair care products, which I evaluated. This is my personal case study. So the structure of diacetate is between the two squares where you can see. This is the structure of the ingredient. And during the synthetic process, if you see, it can lead to formation of certain other components which are impurities, diamide impurity and other acetate, amide impurities. Yeah. So these two impurities, P3, 4, and 5, 6, 7, these are the impurities which were identified. We needed to ensure the exposure of these ingredients, the impurities are safe. And we have one of the starting material, which is amidoamine, used in the production of this lauryl acetate. This is also a known impurity because this amidoamine is a known skin sensitizer. This is a skincare product, meaning it is going to touch the skin. Hair care product, it is going to touch the skin. Therefore, skin sensitization aspects are important. Yeah, because of confidentiality aspects, as this is a real case scenario, I have not included the level of impurities here and there, but the approach, the principle I have just mentioned here. If you look here, peak 5, 6, 7, there are 3 peaks which are bracketed together. We can see the difference is only the difference in the alkyl chain length here at this position, in both positions across all these things. So we considered, or I considered rather, peak 5 as the representative impurity. The reason being, this peak has C11 alkyl chain length on both ends, while others are having higher than C11, either at both arm or single arm. And as per toxicological understanding, based on the experimental evidences and understanding, when the chain length increase, the toxicity decrease. This is a well-known toxicological understanding. So using this approach, I considered this as the most suitable representative. If I mitigate the risk here, it is going to mitigate the risk for other two as well. Likewise, the same here, peak 3 and peak 4, between these two, peak 3 has C11 alkyl chain length here, and therefore it is a representative chemical. That's why I selected peak 3 and peak 5 for this case. Then I looked into the physicochemical properties, everything, the molecular weight and log P 3.75 and 7.73. This Kramer class is one of the criteria which is followed across industry for the components which have no toxicity data. At the same time, the exposure is very low. What is Kramer class? A set of toxicologist scientists, they work together, Kramer and et al., their group, and they published very worthy information where they have looked into various toxicological endpoints of various published data, more than 1,000 chemicals, and they classified the chemicals into 3 categories: class 1, class 2, class 3. Class 1 being more innocuous and class 3 being more hazardous. Based on the data that they have for various endpoints, including organ toxicity and system toxicity endpoints, they proposed if the exposure is less than 90 microgram for class 3 in a day, then they do not pose any risk. Likewise, for class 1, if it is 1800 microgram per day, if the exposure is less than 1800 microgram per day, there is no risk. If the substance is identified, if the chemical is identified to be falling in this Kramer class, then the risk can be mitigated even if there is no toxicity data. So based on the functional groups and the structural features, they arranged all the chemicals and classified. And we used this data, and using the QSAR tools that I described earlier, even here on the slide I mentioned, I used Episuit, Toxtree, and skin permeation calculator. These parameters were calculated. So these tools identified the impurities to be class 3 substance, and there were no structural alert for genotoxicity and carcinogenicity. Even for skin irritation and sensitization, there were no structural alerts. So we were able to mitigate all the endpoints. And the skin permeation calculator calculated value indicated less than 6% to be penetrating through the skin. Based on the theoretical approach, based on the publication by Chorus et al., these chemicals can be assumed to be 10%. This theoretical approach is based on molecular weight and log P without considering the structural features. And based on this, when we try to perform the exposure through the product is applied on the skin, what level it is getting through, etc., etc. The levels were less than 90 microgram per day, therefore we were able to support. And although amidoamine was present there, one of the impurity, it is a known skin sensitizer, but the level was within the reported acceptable level. Many a time, based on the publications, we already determine what is the level which is acceptable, some 0.1%, 0.05%, whatever levels are. So based on that, it was considered acceptable. Overall, we mitigated the risk using this in silico tool in this manner. Where we look for toxicology data, like that is another important piece. If we get all the data easily, then we know what is a descriptor to be used, NOEL, OL, etc. But if data is not available, we hunt for data. We search in different sites. I have provided link to the sites in the abbreviation on the screen. So these are various sites. I don't want to spend time here, but most of the folks on the call, they may be already, who are involved in research, maybe using or maybe touching upon. One of the site is PubMed. Certainly people may be using that. Some of the site is like CheckFA and EFSA, which are food authorities based information. ECA is European Chemical Agency. EPA is US EPA. NTP is National Toxicology Program from US Health Agency. EMA is European Medical Agency. These are different websites where we can just look into for any known chemical. Since my work primarily is in the OTC activities, occasional new chemicals I have handled, but oftentimes the work is primarily based on existing data, either internally generated by J&J or from the database which is cited here. And for the new chemical, again, the data sets requirements are the same as it is mentioned this previously. And what is important, there are well laid down guidances which are available for all this information. OECD guidance, US EPA guidance, those are all available. People can just look in there for what endpoints to be considered within a study. When you plan a study, just make sure those things are covered so that you are at the end maybe using animals for the study, if you are planning for animal studies. And people are very sensitive. This is one of the important learning which I have learned over a period of time. My days as a student, graduate student, and my association with Indian industry, when I moved to Johnson & Johnson, the mindset is totally different. Even today, if I have to use few animals, 3-4 animals also, then there are a lot of restrictions of using animals. The justification, rational, it has to be fully laid down why we need that animal experimentation. Unless otherwise that is justified, do not perform animal experimentation unless otherwise it is needed. So my key learning, Dr. Anita already covered some of the things. Networking, collaboration, these are very important in the industry as well as academia, wherever you are. Please participate in various training programs, workshops, webinars, and other things. That is where we can learn a lot. Being a student, BCP provided me a lot of opportunity to learn by way of volunteering in those events, took part in those activities, learned many things, and also participated in various webinars or maybe workshops, training programs, and participated. That helped me a lot. Even you also think on updating your knowledge. That is very, very important. Book and what we do at academic level is important. This gives a lot of basic knowledge, understanding. And as we move into industry, the focus is too specific, not as wide as academia. I'm missing those aspects. We have liberty to plan, design, do various things in academia. Utilize those opportunities when you are there in academia. Talk to your supervisor, convince, take alignment on your experimental designs. This is what I am thinking. This is how I'm going to perform. These are the rationale I'm laying down why I'm performing those studies. Take alignments, and everyone may be facing different adversities during the phase. Even I have faced, but those are the stepping stones. How to fix those conflicts. Think on those aspects. The conflicts can come in different ways. As I said in the beginning, my lab manager at my previous industry was not ready to accept my proposal. So I have to work upon that and then convince. So unless you have the ability to convince people, that by way of communication, which Dr. Anita reiterated multiple times, communication is very important. And how do you communicate? That is very important. It should be in a convincing manner and with end in the mindset. What are you trying to achieve? What is the objective? What is the purpose of it? When you are trying to justify those purpose, you will be able to convince nicely. Work out a couple of times. What are you going to talk to people? Be it your supervisor, be it your external stakeholders, whoever it is, colleagues, any other cross academic persons or industry person. What are you going to talk? Work on that. Ask yourself questions. Ask question repeatedly why we are doing. I was never satisfied. Believe me, I have learned so many things in BCP during my experimentation only by asking repeatedly questions. I can narrate various stories. When I was performing isolated heart experiment in BCP, I have got fantastic heart rate responses. I was very happy showing everyone that I have isolated a compound or fraction. This was showing beautiful activity. Again, this is a real-time example to everyone. I went and discussed with Dr. Saraf as well. And he also said, yeah, it is very nice. You are showing wonderful activity. The chemical which I have isolated then was showing proper dose response curve for increase in heart rate. It was fantastic. I was having sleepless night in those happy moments. This is a reality. But I asked repeatedly questions. Am I doing something correct? Repeated the experiment. I got the same results again. But I looked into literature later. This is where I was wrong, because I was isolating the component using a TLC plate. So the TLC plate was prepared using that calcium sulfate, whatever the material is, that coating. I scrapped everything and then extracted out using a solvent. The solvent, of course, I used methanol. I remember all these things. I can narrate very well because those are green memories always. Extracted out and evaporated the solvent and went and then performed the experiment. Experiment was beautiful. At the end, I realized it is false positive in the experiment. Everything because it was having calcium sulfate as a binder. TLC plate was having calcium sulfate as a binder. That trace level of calcium which leaked into my extract gave me positive results. Then I myself went back and told Dr. Saraf that, sir, this is a false positive. I cannot use this result. I have learned something. So there are a lot of learning in your day-to-day experiments. Look for non-obvious things. Obvious things you will always see. Don't get contented with what you are doing. Take additional steps to make sure that it is flawless. Make sure you are addressing all the queries that come to your mind. Keep asking questions to yourself repeatedly. That's what I would say. And you would see a fantastic outcome in your thought process with respect to technical things, and that will hone you up for your future in your career. There are two things important. One is how do you enhance your technical skills? Two skills, in my opinion: technical skills and soft skills. Technical skills, keep doing good work, ask questions yourself at every step that you are doing. But technical skill alone is not going to give you something which is good for you in the career. Meaning, like, if you want your career to be steeply growing, soft skill is equally important. If you are a good technical skill person, you will be good for benchtop work. That's how most people, I'm very openly saying this industry trend or practice. If you have both qualities balanced, technical and soft skill, the presentation, how do you identify the analytical skills? How do you identify the areas? Krishna sir is here. He is one of the role model for me to improve my analytical skill, along with Dr. Vadlamudi Rao. So learn from them. BCP has very high talented professors who have, who have been instrumental in changing me to this level. So I ask questions, and some of the professors here would be knowing that even during any seminars, I used to ask questions, because asking question is one to learn, another to coach. Coaching is by way of asking questions, so that the other person should not feel offended. They should go back and then see what is still missing. They can update. That is very important. So keep asking your colleagues, your friends on what they are doing. So in a way, you are indirectly coaching them. So another important piece which Dr. Anita said, industry is not working in silo. So people are working as team. We have to work as team together at every level, even at academic level as well as industry level. When we do that, certainly success is your very reachable goal. That's all from me. Thank you.
Thank you, Suresh sir. Thanks for a wonderful presentation. It was very interesting and very elaborated presentation, where you have shown your journey and how you learned toxicology from BCP, from your mentors like Dr. Saraf and Dr. Rao. And you have mentioned about the importance and role of toxicology in drug development. You have also touched on small critical points in toxicology studies in drug and cosmetic development. You mentioned various toxicology studies and their endpoints, dose response studies, and how the risk assessment is done, and the in silico, in vitro studies for cosmetic and personal care products, and very importantly, the very interesting case study of in silico method. And in the end, the career path, your career path you have described, what hurdles and how the achievements come after the struggle. And in the end, balance between how to maintain balance between technical and soft skills. And thank you so much for a very interesting and elaborated presentation. And if anyone has question, they can ask. And I have one question for you, sir. That as we all know that Indian regulation is more lenient compared to Western world. So how do you see its evolution in future?
India is not lagging behind, I would say. That's what already India has banned animal experimentation for cosmetics. India is growing, and we are the people who are going to change Indian regulations, perspectives, everything. And I'm sure we have right talents within Indian community, be it from academic teaching professors, the students, everyone. When we move out, when we are in the place to shape the regulations, then we need to take those initiatives. Currently, I believe certainly there are several people who are already taking the regulations. That's why the ban for animal studies had happened in India. Likewise, I can say the other thing. India has banned the use of single-use plastics like Western world. So there are various things India is coming up in terms of regulations and other things. While we cannot define certainly a time frame by when this is going to happen, but we are certainly on the path. We are learning from Western world with the advent of technologies, the communication technology, information technology, and everything. The barriers are open across globe. So adapting the trend is very important. The reason is very simple. If Indian companies want to do business across globe, then they need to adopt these standards. It is in one word, right? When Indian industry is adapting this, they are going to drive the regulators as well by shaping the regulations. So these are win-win situations which are likely to happen. The markets are getting expanded. The world is looking at India, China for growth opportunities. Western worlds are totally saturated. So these Asian countries are looked at for market expansions and other things, which I believe everyone would agree. Even from J&J perspective, this is a realistic scenario. So as they are doing that, yeah, the regulations will be more or less same across globe, I would say. I would not say it is very lenient even currently.
Yeah.
So, Suresh, if I may add to, I absolutely agree to what you say. But also saying that since I am now in the regulatory environment and stuff, I would here argue that that the statement that India is lenient and more with their regulations, that's not true. If you see companies like Cipla, etc., who are indigenous companies, and the market, as you rightly mentioned, is also global for them. And they follow the regulations. Of course, regulations are varied. There are attempts to harmonize it, but India also has strict regulation when it comes to importing chemicals into the country. Yeah, there's something called the Bureau of Indian Standards, where even a company like Merck, if they want to get their chemicals into India to sell further, which you use in your labs, we have to fulfill the Indian requirements with inspections, GMP approaches, and all those documentation is required. So it's not that the Indian regulations are lenient than the European or the US. Of course, we don't do path-breaking regulatory research, I can say that. Or let's say the nitrosamine issue. I'm sure, Suresh, you must have also come across that. It was a big blow for the industry where all of a sudden, all the projects went on the side and we all were looking only at nitrosamines. Like, is it there in my product? Will it affect? And you have to give. So such kind of things, the reaction time in the European and the US regulations are faster, I would say. And India is getting there. Like, we are thinking of once Indian authorities will also move into the ICH. We follow the ICH guidelines, but all coming together, this will all, then we are hoping that would be one ideal day when everything is harmonized and we don't have to keep changing the rules. But I totally agree with what Suresh said. But that being said, it's not that India is behind. We are on the right track and in the right way.
Yeah, just to add on, Anita, thanks for touching upon nitrosamine impurities. That's one of the impurity, and the world is looking at it. Every pharma industry is looking at whether they have nitrosamine, and they have to comply, at least in the Europe and US, everywhere. They have to comply the regulations for nitrosamine impurities. That said, when you are in that business, say for example, ranitidine, Indian government, although it is not imposed, it is an implied message across industry that they have to control that impurity because it is a known carcinogen. And nobody wants to market a product which is having an impurity which has carcinogenic risk, right? So that's where the industry will take a lead in some case. And as industry takes a lead, they are going to shape the regulations slowly to make sure this happen. At some situations, industry and governmental agencies can come together. This also happens several times. They work in partnership. They define the criteria of requirements, and eventually they lay down the guidances which is applicable regionally. So they will come up even for nitrosamine in this case. It's very important impurity aspect which currently even I have personally worked on various nitrosamine impurity qualifications.
Thank you so much for your answer and the presentation. Now I will request Mala Madam to give vote of thanks. Over to Mala Madam.
Good evening, everybody. Once again, it is a pleasure to be delivering the vote of thanks when two of our great achievers among the alumni, Dr. Anita and Dr. Suresh, have delivered the talk. Many thanks to both of you for having agreed in the first place at a very short notice. And also there was a change in the date. I think initially some date was fixed. Later on the date was modified. So there also you have, just in one message, you have agreed to the change in date. So thanks for agreeing and for the wonderful presentations, elaborate presentations where you have started from your college days to each step in your career. Anita has really spoken about the various challenges and how the various skills that are required for the students as they move up from the university to the industry. Suresh has also highlighted the various steps from animal experimentation to in silico or benchtop toxicologist. I think I've given very good examples, and both of you have emphasized on the multiple skills that a student requires and the lifelong learning. So thank you very much. Also, many thanks to all the people who have attended, the alumni who have attended, the teachers who have attended. Thanks to the committee members, Dr. Parizad, Ashish, Agnivesh, and Shruti for having looked into the various activities and conducting this. Thanks to Shrey and his team for the website arrangements. Thanks to one and all. Okay. So I think till the next Chai Pe Charcha, I think we will, we hope we will see all of you again in the next Chai Pe Charcha. Thank you.
Thank you, ma'am. Thanks for inviting us for providing this talk. Yeah, thank you very much. Thank you, everyone. Yeah, BCP is like a home to me, and although I have not visited the recent days, it was a home to me, and many of you know that. I have my attachments to BCP. At the outset of this meeting, there was a request, to provide support to some of the students. Please provide or share those details. How to?
Yeah.
So I will consider that.
Are you members of the alumni association?
Yeah, that is one thing. Which,
that's one more thing we want to be.
You can register on our website.
Okay.
Go to
and become members, and also encourage your colleagues, whomever you know, to
Sure,
members.
sure.
So you will have all this information directly. We put it up on our
Okay.
website. Yeah.
Okay.
Yeah.
Thank you. Thank you,
so
ma'am.
much.
Then,
Thank you. Thank you.
Yeah.
Bye-bye.
Thank you very much. Bye-bye.
yeah, bye. Thank
Have a
you.
nice day.
Bye-bye.
Bye-bye.
Have a nice day and week.
Thank you.
Thank you.